My name is Bill Catterall. I'm a professor of pharmacology at the University of Washington, and I'm really pleased to participate in the iBiology lecture series today. Our topic is electrical signaling, life in the fast lane. We use that title because electrical signals are involved in all of the really rapid processes that humans and other animals participate in. For example, electrical signals are used to communicate, to encode and process information in the brain, to tell your muscles to contract at the right time, and in many other rapid processes. So to think about how fast biological processes need to be, we can take a look at this example. Here we have a local hero here in San Francisco, Buster Posey, hitting a fastball, and that fastball left the pitcher's hands. Buster picked it up by his vision, decided where it was going to cross the home plate, swung the bat, and he did all of that in much less than a second. So how can he respond so fast? What type of signaling is important to let this happen? So here on this slide, I illustrate the different time scales of biological processes. So for example, at the top of the slide here, you can see it takes almost a day for DNA replication and cell division to take place. Many kinds of processes that take place in cells require almost an hour for gene transcription and to make a large protein, for example. Hormones might act on the time scale of minutes, and typical enzyme activities take place in the time range of 1/10 of a second to a second. But the really fast processes, electrical signaling, takes place on the millisecond time scale, 1,000 times as fast. And that includes vision, hearing, nerve conduction, and muscle contraction. All of those things that Buster Posey was using in the last slide in order to hit that fastball. Here's an example of encoding information using electrical signals. This is from a classic piece of work by Hartline in the 1930s. He was shining light on the eye of a horseshoe crab and measuring the electrical signals that the eye sends back to the crab's brain. And as this slide illustrates, he found that the frequency of electrical signals, shown here on these drawings, increased as the light intensity increased. So 10 times higher light intensity gave a 10 times faster rate of electrical signals, and 100 times faster gave even more electrical signaling at a faster frequency. This illustrates a key principle, which is that information is encoded in the frequency of electrical signals, both the intensity of light, the force of muscle contraction, and many other physiological properties. So how do cells make electrical signals? They have no batteries, they have no wires. Cells use ions to make electrical signals. Ions are small charged particles, and in this case, they're charged atoms, sodium and potassium ions. And to make an electrical signal, the cell first opens up a channel that allows sodium ions to move into the cell. As they move into the cell, the sodium ions bring a positive charge into the cell with them, and they change the membrane potential across the cell, as shown here in phase 1 of the action potential. The cell becomes more positive, and we say the cell membrane is depolarized. Then, very shortly after that, within a millisecond, potassium ions leave the cell through specific potassium channels. And potassium ions leaving the cell are responsible for this second phase of repolarization of the action potential. And both of these events take place within 1 millisecond. These specific movements of sodium and potassium across the cell membrane take place through ion channels. And so much of our discussion today will be about exactly what the ion channels are and how they work. And it's really the work of Hodgkin and Huxley from the 1950s that shows that moving ions make electrical signals in nerve and muscle. So this is a more scientific presentation of the same kind of information taken right from the classic Hodgkin and Huxley papers in 1952. And they worked in the squid giant axon, and they showed here that the membrane potential change in the squid giant axon, marked EM here, goes from a resting membrane potential of about minus 65 millivolts. During an action potential, it goes all the way to plus 20 millivolts. It crosses the 0 millivolt line and then returns back to the resting membrane level at the end of the action potential. And they showed that sodium channels are responsible for the rising phase of the action potential. They open and let sodium into the cell, as we saw. And they also found that sodium channels have two gating mechanisms: activation, which controls their opening, and inactivation, which causes them to close within a millisecond after they open. Potassium channels open more slowly. They let potassium out of the cell, and then they repolarize the cell membrane, as illustrated here. In a nerve fiber or a muscle fiber, as illustrated on the bottom of the slide, the action potential begins in one place and then moves along the fiber. And it does so because it depolarizes the next bit of membrane to move ahead and conduct at a rapid rate. So when you move your arm, or Buster Posey swings his bat, an action potential moves all the way down from your spinal cord down to your hand and to swing the bat, and it does so in a very short period of time, much less than a second. So scientists have found ways to actually measure the sodium current moving through one single sodium channel molecule. And this is a major advance in our field, made around 1980. And the way that experimenters found to do that is to capture a small bit of membrane, as shown here, in a recording pipette. Such a small bit of membrane that there's only one or two sodium channels in it. Then, when you stimulate the membrane with an electrical current, you can see, as the slide shows here, openings of one or two sodium channels. And these different rows on the slide show different repeated stimulations of this little patch of membrane, and each time one or two sodium channels open. If you add up the little sodium currents that are appearing each time one or two sodium channels open, on the bottom of the slide here, you would record a sodium current similar to that seen in the cell. Sodium currents are always drawn downward, as you see here, as inward current is a negative quantity by convention. It's interesting to compare the size of these very tiny sodium currents to other electrical currents that we think of in our home, for example. So a single sodium current is about 15 picoamperes. That's about 10 million sodium ions per second, so it's a lot of sodium ions that are going through the sodium channel each second, but it only generates 1/1,000,000th of the sodium of the electrical current that you would find, for example, in your household outlets, typically 15 amperes instead of 15 picoamperes. So sodium channels make very tiny currents, but they add up to give the action potential and create the electrical signals that allow this rapid form of cellular communication to take place. Many animals make toxins that target sodium channels to paralyze their prey. And I bring that up now because, as you'll see, scientists have used the toxins as tools to study the sodium channels. So on this slide, we see a number of nice and in some cases rather fearsome looking animals that make toxins that act on sodium channels, and they use them to paralyze their prey. Particularly interesting one is the cone snail. These snails eat fish. Think of the problems that a snail has if he dines on fish. The snail stings the fish with an extensible proboscis here that contains a tooth, and the fish has to be paralyzed instantly. If it swims even one stroke, dinner is gone. So the snail has unbelievably potent and fast-acting toxins. So in experiments that were done in my lab many years ago, we used toxins from the scorpion, illustrated here, to find the sodium channel molecule. So to start, I've just shown on this slide what the toxin of the scorpion does to the function of the sodium channel. So the left side of this slide, again, shows the sodium currents that you would record in a nerve cell that was firing an action potential, and sodium channels were activated. So here on the trace that's labeled control, you can see the normal fast activation going downward, and then inactivation of the sodium current. The scorpion toxin slows the inactivation process, so the sodium current lasts longer. And if you put in more toxin, it slows it even more, so the sodium current lasts even longer. And we can add up experimental results from many different concentrations of toxin and generate this curve that illustrates how the toxin increases the time of the sodium current as we add more and more toxin into the experimental preparation. The prolonged sodium current paralyzes the nerve by depolarizing it all the time and preventing it from generating action potentials. So as I mentioned, we used scorpion toxins and other toxins as a way to find the sodium channel molecules in the brain. So how can we discover the sodium channel protein molecules in the brain? There are more than 20,000 types of proteins in the brain. We'd like to be able to recognize and isolate just one of them. Unlike enzymes, which often are active once you take them out of a cell, the sodium channel really has nothing to do when you take it outside the cell. It has nothing to transport sodium across. So we wondered, could we extract all of the molecules from the brain and then catch the sodium channel by binding it to a toxin? It's a little bit like fishing with the scorpion toxin as a bait. So here we have a nice mountain lake, and you have to imagine that's actually a beaker containing all the proteins from the brain. And we're going to throw in our scorpion toxin bait and try and hook it on to the sodium channel protein. And we were lucky. The scorpion toxin bait found the sodium channel, stuck to it very tightly, and allowed us to catch the sodium channel protein and reel it in and separate it from all the other 20,000 molecules that are present in this case, in the brain of the rats that we did these experiments on. So looking more closely at the sodium channel, it's really well designed. It's a molecular machine for electrical signaling. So if you focus on the cartoon part of the slide first, you can see the sodium channel, it consists of three components, or subunits as we call them. The alpha subunit, here highlighted in a red or orange color, is the large subunit. It forms the pore of the channel that lets sodium move through, and it's also the target for the various toxins, including scorpion toxins, that act on sodium channels. And the alpha toxin is a protein of about 260,000 Daltons, shown on the left side of this slide in a SDS gel that separates proteins by size. It's the protein band on the top. The alpha subunit is associated in nerve and muscle membranes with smaller beta subunits, and here the examples of the beta 1 and beta 2 subunits are shown in the cartoons. We call them auxiliary subunits because they're not involved in the direct function of the sodium channel. They don't conduct sodium or respond to voltage. But they're very important because they regulate sodium channel function, and they help to locate sodium channels in the right places in cells, which is really important. On the lower left quadrant of the slide, you can see the recording of a tiny sodium current, a 15 picoampere sodium current, from one single, now purified sodium channel molecule, separated from all the other molecules from the brain. And by recording those currents from the purified sodium channel molecule, we could persuade ourselves we really had purified the right protein because it had the right size and the right voltage dependence of sodium currents. So our laboratory and many other laboratories in our field have worked over many years to try and learn what parts of the sodium channel protein are involved in its different aspects of its function. And so this slide illustrates some of that work. So the diagram shows the sodium channel protein in a two-dimensional map. All proteins are made of a long chain of amino acids. And in the case of the sodium channel, the chain of amino acids is 2,000 amino acids long, from the amino terminus on the left to the carboxy terminus on the right. And the protein threads back and forth through the membrane 24 times. And so we say that it has 24 transmembrane segments. Those transmembrane segments are organized in 4 groups or 4 domains. You can see one of those domains here. Each domain has 4 transmembrane segments that are called S1 through S4, which are involved in voltage sensing. And they have 2 transmembrane segments that are highlighted in green here, S5 and S6, and a small P loop in between them. And that's the part of the channel that forms the pore. So the sodium channel has 2 functionally separate modules, the voltage sensing module, S1 through S4, and the pore module, S5 and S6. As I mentioned on the earlier slide, in which I was illustrating the classic work of Hodgkin and Huxley, sodium channels open within a millisecond, but then also inactivate within a millisecond. The fast inactivation process takes place right here in this little piece of protein that is on the intracellular surface of the sodium channel. We call that the inactivation gate, and it's responsible for closing the sodium channel and bringing the membrane potential back to normal within 1 millisecond after opening of the channel. So the first part of the sodium channel that we really understood at the structural level was the inactivation gate. And so we see on this slide a little more detail about how the inactivation process works. So the top part of the slide is a cartoon, and it shows the 4 domains of the sodium channel surrounding the central pore, and it shows the inactivation gate folding into its binding site at the intracellular mouth of the pore during inactivation. So when the channel opens, the inactivation gate is in its open position, as on the left part of the slide. Then, within a millisecond, the channel inactivates, and the inactivation gate closes over the intracellular mouth of the pore, and here binds and blocks sodium movement. So we were able to produce the inactivation gate protein separate from the rest of the sodium channel by itself and determine its structure using the technique of nuclear magnetic resonance, or NMR. And as you can see, the inactivation gate has a very rigid alpha helix, and then in a 2 turns of the polypeptide chain, the inactivation gate has 4 key amino acid residues: an isoleucine, or I, a phenylalanine, or F, a methionine, or M, and a threonine, or T. These amino acid residues are the latch that hold the inactivation gate closed. You can see them here. I, F, M go in and just bind to the intracellular mouth of the pore, block the sodium movement, and end the action potential. So this slide is a little movie, and it will illustrate for you how the inactivation gate works in real time, but in really slow time compared to the normal action potential. It also shows 1 voltage sensing module here on the left, and the pore module in the center in red. A real sodium channel would have 4 voltage sensing modules, but we can't draw them all because it becomes very confusing as I run the movie. So when we run the movie, we'll see that the voltage sensing module here undergoes a change of shape conformational change in which these little blue balls, which are positively charged amino acid residues, move outward. The change of shape, the conformational change in the voltage sensing domain, pulls on this connecting linker here, and the connecting linker pulls open the pore and allows sodium ions, illustrated as green balls on the top of the slide, to move through, and then very soon after the sodium ions move in, the inactivation gate closes. So we'll see if we can run this movie now. There you go. So each time your nerve or muscle cell fires an action potential, your sodium channels are doing exactly that. Sodium channels are important in another way. They're the receptors for important classes of drugs. One that you're very familiar with is the local anesthetics. Local anesthetics prevent pain by blocking nerve sodium channels. For example, when you go to the dentist, as this lady is doing here, the dentist will give you an injection of a local anesthetic, usually lidocaine. And it's really good that that is an effective drug, because then your brain doesn't know about the bad things that your dentist is doing in your mouth. Not only local anesthetics act on sodium channels, but also drugs that are used in the treatment of epilepsy, in the treatment of cardiac arrhythmia, and in the treatment of chronic pain. All of these conditions are caused, at least in part, by firing of too many action potentials, too many electrical signals, and so sodium channel blockers are effective treatment because they reduce the firing of electrical signals. So the sodium channel blockers work in a very simple way, and that's illustrated in cartoon form on this slide. As you can see, the local anesthetic molecule is thought to enter and block the pore of the sodium channel. It goes right in from the intracellular side here, binds to a binding site, and since it's there and blocking the way, the sodium ions can't move through the pore, and the action potential is prevented. We actually know quite a lot about how the local anesthetics bind to their receptor site on the sodium channel and block the sodium channel, and that's illustrated on this slide. So first on the left, this is the same drawing that you've seen before, and here I just want to emphasize that the drug molecules bind to the pore lining segment, the S6 segments of the sodium channel. The right side of the slide, 3 of those S6 segments are illustrated, and they come together to form the closure of the pore at the intracellular end. And this slide illustrates the S6 segments from domain 1, labeled 1S6 in red, from domain 3, labeled 3S6 and in green, and from domain 4, labeled 4S6 and in blue. So in the slide, you see the yellow molecule here is a local anesthetic, and the amino acid side chains with which it's interacting are shown in this space-filling format. So here in 3S6, here in 4S6, and here in the 1S6 subunit, 1S6 domain, pardon me. So the local anesthetic binding site then is right in the pore. When the drug binds there, the pore is locked shut. The drug blocks the sodium permeation pathway, and these are very effective drugs in therapy. As I mentioned, not only local anesthetics are important among sodium channel blocking drugs, but also anticonvulsant drugs, for example, phenytoin or dilantin, carbamazepine and lamotrigine, widely prescribed for treatment of seizures and other conditions, and antiarrhythmic drugs prescribed for cardiac arrhythmias, including procainamide, lidocaine, mixilotine, quinidine, and flecainide. So the sodium channel then is both physiologically important and pharmacologically important as a target of important therapeutic drugs. So I'll just finish my lecture with this movie again, and I'll try and reiterate some of the major points that I've made. So first, electrical signaling is fast, and everything that we do quickly depends on electrical signals. Electrical signals are generated by the movement of sodium and potassium ions across the membrane, and that movement is mediated by ion channels. We've discussed the sodium channel today, but there is an equal body of information about potassium channels as well. The sodium channel is a rare membrane protein in a nerve cell or in the brain, yet we were able to find it using specific toxins as molecular bait to fish it out of the extract of proteins from brain. And that sodium channel has two functional modules that are shown here. First, on the left, the voltage sensing module, which responds to changes in membrane potential, undergoes a conformational change and pulls open the pore. Sodium ions quickly rush in to give the depolarizing phase of the action potential. Then the inactivation gate closes to stop the sodium movement, and then potassium channels repolarize the cell. So all of this will take place now when I push the button, and you can see the action potential for the last time, at least in this talk. I'll move to the last slide, which is just a blank one, and say thank you very much for your attention, and I hope you'll come to another talk at iBiology when you have time. Bye now.